welcome to this explainer. Today, we're diving straight into the hidden microscopic mechanisms that literally govern human health, cellular balance, and the absolute cutting edge of biotech. We're talking about allosteric regulation. Now, I know it sounds like a heavy, intimidating biochemical term, but stick with me, because by the end of this, you're going to see it as one of the most elegant, fascinating systems in all of nature. I mean, these are quite literally the hidden switches of life, and they're operating millions of times a second inside of you right this very moment.
It's a pretty wild question to think about, right? How exactly do your cells know when to stop making energy? Your body is essentially this massive, impossibly complex factory. If it just kept producing energy in molecules nonstop without a brake pedal, it would just burn through all your resources and, well, destroy itself. It's a massive logistical nightmare. So there has to be a way to communicate supply and demand on a purely microscopic level. It requires this highly evolved, elegant solution to maintain perfect balance or homeostasis. And that beautiful solution is exactly what we're decoding today.
Okay, let's dive into this. Here's our roadmap for today. First, what is allosteric regulation? Then, the mechanisms of enzyme control, followed by feedback inhibition in action. After that, we'll look at models of enzyme cooperativity, and finally wrap up with medical and biotech applications.
All right, section 1, what is allosteric regulation? Before we get into the nuts and bolts, let's establish our foundational vocabulary. Let's break this definition down nice and clearly. Allosteric regulation is the control of enzyme function by the binding of an effector molecule to an allosteric site, which causes a conformational change that affects enzyme activity. Okay, what does that actually mean? Think of this effector molecule, sometimes called a modulator or regulatory molecule, kind of like a remote control. Hopefully one that doesn't get lost in the cellular couch cushions, right? It doesn't actually need to be right inside the main engine of the enzyme to turn it on or off. It operates the enzyme from a completely different location, essentially shape-shifting the enzyme from a distance. So notice the stark contrast here between the two main sites on an enzyme. Over on the left, we've got the active site. This is the catalytic region, where the substrate binds and the actual chemical reaction happens. Think of the active site as the conveyor belt in a factory machine. You know, it's where the actual heavy lifting gets done. But on the right, we have the allosteric site. This is the regulatory region. It binds those remote control regulatory molecules we just talked about to control the enzyme's overall activity. So if the active site is the conveyor belt, the allosteric site is the wall switch all the way across the room that powers the belt up or shuts the whole shebang down. Moving on to section 2, mechanisms of enzyme control. Okay, so we know we have a conveyor belt and a wall switch, but how exactly does this remote control actually manipulate the enzyme's physical structure? Let's walk through how this incredible mechanism builds step by step. Step 1, the regulatory molecule docks perfectly into the allosteric site. Step 2, this binding event triggers a physical ripple effect across the entire protein. The enzyme literally undergoes a conformational change. It shape-shifts. Step 3, because of this brand new shape, the active site, somewhere else on the enzyme, is altered. It becomes either more or less suitable for its substrate. And finally, step 4, as a direct result, the overall activity of the enzyme either surges or drops. It is an incredibly precise, physical cascade. Now, this molecular switch goes both ways. On one side, we have positive regulation, or activation. This is where the effector molecule acts like a gas pedal. It binds, stabilizes the active shape of the enzyme, makes it easier for the substrate to bind, and boom, enhances the reaction rate. On the flip side, we have negative regulation, or inhibition. This is your brake pedal. The molecule binds, twists the enzyme into a much less accommodating shape, reduces substrate binding, and successfully slows the whole metabolic pathway down. That brings us to section 3, feedback inhibition in action. This brings us to arguably the most crucial real-world application of that brake pedal we just talked about, feedback inhibition. This occurs when the final product of a metabolic pathway actually turns around and inhibits an enzyme involved in an earlier step of that exact same pathway. Imagine a cell is building an amino acid, step by step, and suddenly it has enough of it. That final amino acid acts as the negative regulator. It goes all the way back to enzyme number 1 and hits the brake. Think of this exactly like your household thermostat. When the room gets warm enough, the heat itself tells the furnace to shut off. It's brilliant. It prevents the cell from hoarding unnecessary resources and wasting precious energy. And this brilliantly illustrates how the whole thing plays out using one of the most important enzymes in your body, PFK1, which manages glycolysis. That's the breakdown of glucose for energy. Just take a look at the top row. When cellular energy is low, molecules like AMP and ADP start to build up. They act as activators. They bind to PFK1 and basically say, hey, we are starving. Step on the gas, increase glycolysis. But check out the bottom row. When you have high energy, ATP and citrate are abundant, and it's actually amazing when you think about it. ATP itself acts as an inhibitor. It binds to the allosteric site of PFK1 and says, whoa, we have plenty of energy, hit the brakes, slow glucose breakdown. It manages the pathway purely based on the cell's immediate energy levels. Up next, Section 4, Models of Enzyme Cooperativity. If we zoom in slightly to look at the physics of this binding, we find that these enzymes are often complex machines with multiple subunits that have to communicate internally. Scientists have developed a few models to explain how these invisible interactions work, specifically when one subunit changing shape affects its neighboring subunits. There are two primary academic theories here. The concerted model, also known as the MWC model, argues that the entire enzyme exists strictly in one of two states, meaning all subunits change together at exactly the same time. Conversely, you have the sequential model, proposed by Koshland, which suggests more of a domino effect. It argues that the binding of a molecule causes gradual changes, where the subunits change shape one by one. To clarify the concerted model specifically, researchers refer to these two strict states as the T state and the R state. T stands for tense. In the tense state, the enzyme is rigid. It has lower activity and a much lower affinity for its substrate. But when an activator binds, the entire enzyme elegantly toggles into the R state, which stands for relaxed. In the relaxed state, it has higher activity and easily binds to its substrate. It's this constant microscopic toggling back and forth between tense and relaxed modes.
Which leads us to our final section, Section 5, Medical and Biotech Applications.
All right, let's zoom all the way out to the macro level. Why does mastering these microscopic switches actually matter to us? Well, because modern science is actively hijacking these natural remote controls. In drug discovery, pharmaceutical companies are now designing drugs to specifically target the allosteric site rather than the active site. Why? Because it offers much greater specificity and far fewer side effects, mostly because the drug doesn't have to compete with the body's natural substrates. On top of that, we are engineering these enzymes for industrial biofuels, and in synthetic biology, scientists are programming entirely new cellular behaviors just by manipulating these metabolic switches. It is literally a game changer.
But, we also have to understand the dark side of allostery. What actually happens to the human body when these critical switches get stuck in the on or off position? In cancer, for example, malignant cells often modify enzyme regulation to literally turbocharge their metabolism, a phenomenon known as the Warburg effect. They basically lock the gas pedal down to feed their rapid growth. Additionally, severe metabolic disorders occur when these regulatory enzymes are defective. This leads to either a really dangerous accumulation of toxic metabolites or a catastrophic reduction in cellular energy. To combat these diseases, the tools scientists are using today are absolutely mind-blowing. We are visualizing these shape-shifting proteins using cryo-electron microscopy and X-ray crystallography. We're using massive supercomputers for molecular docking simulations. And looking to the future horizon, artificial intelligence is now designing brand-new allosteric drugs completely from scratch. We are entering an era of precision-targeted cancer therapies where we can selectively turn off the mutated enzymes feeding a tumor, all without harming the healthy cells. No way, right? It's unbelievable. Which leaves us with a pretty fascinating thought to wrap up on. We've gone from just trying to understand how a microscopic enzyme shifts its shape to literally using artificial intelligence to build custom remote controls for human biology. So if we can perfectly master the molecular switches of life, what diseases will we be able to turn off next? Thank you so much for exploring these incredibly complex yet beautiful systems with me today. Keep wondering, keep learning, and I'll see you in the next explainer.